Every living organism on Earth, from a microscopic amoeba to a blue whale, is constructed from incredibly small, highly active functional units called cells. For most of human history, this entire foundation of life was completely invisible to us. The unaided human eye has a strict resolution limit of about 0.1 millimeters. Anything smaller than a grain of sand simply blurs together. Because of this optical limit, we had no idea that life thrives in environments that seem completely uninhabitable. In the Puga Valley of Ladakh, hot springs reach nearly the boiling point of water, yet they are teeming with heat-loving unicellular bacteria called thermophiles. It wasn't until 1665 that Robert Hooke used an early microscope to look at a thin slice of cork. He observed tiny box-like compartments and named them cells, providing the first glimpse into biological architecture. This observation evolved into the classical cell theory. Scientists realized that all organisms are made of cells, and, as Rudolf Virchow later stated, every single new cell arises from a pre-existing cell. Understanding biology meant we first had to invent the optical technology to break past our own sensory boundaries and observe these foundational blocks directly. Once we could see inside these structures, biologists realized that all life forms fall into one of two primary architectural designs, defined entirely by how they package their genetic material. The older, more primitive design is the prokaryotic cell, found in bacteria. They lack a defined membrane to hold DNA. Instead, their genetic material floats loose in a central nucleoid. Plant and animal cells use the eukaryotic design, securely enclosing genetic instructions inside a well-defined membrane-bound nucleus and organizing internal tasks into specific organelles. Enclosing the DNA and partitioning tasks into separate compartments provided a massive boost in cellular efficiency, paving the way for large, complex, multicellular organisms to evolve. Every cell, whether prokaryotic or eukaryotic, protects its internal machinery with a selectively permeable plasma membrane. It operates as a fluid mosaic, a flexible double layer of lipids with proteins embedded throughout. These proteins act as selective gates, letting specific nutrients inside while blocking others. This membrane is highly sensitive to its environment. If a cell is placed in a hypertonic solution, meaning the liquid outside has a higher concentration of solutes, like salt, than the inside, water will diffuse out of the cell across the membrane. This process, called osmosis, causes the cell to rapidly shrink. Traditional Indian food preservation leverages this exact biological mechanism. By packing pickles and murabbas in high concentrations of salt or sugar, they create a severe hypertonic environment. Any spoilage bacteria that land on the food quickly lose their water through osmosis and die of dehydration. When this fluid loss happens in human cells, it triggers a medical emergency. Diseases like cholera cause the intestines to secrete massive amounts of water and electrolytes, leading to severe, often fatal dehydration. To combat this, Dr. Dilip Malanabis helped popularize the oral rehydration solution. By providing a highly precise ratio of salt and sugar, ORS relies on specific solute gradients in the gut. The body absorbs the sodium and glucose, which then forces water back into the severely depleted cells through osmosis. Understanding how to manipulate the concentration of solutes across microscopic cellular borders provides a direct mechanism to control bacterial growth and save millions of lives. Inside a eukaryotic cell, the semi-fluid cytoplasm acts like the floor of a sprawling manufacturing plant, with every action coordinated by DNA instructions stored inside the nucleus. Tiny structures called ribosomes synthesize proteins based on those instructions. These proteins are pushed into the endoplasmic reticulum, a network of tubes that manufactures lipids and packages these new materials for transport. The materials travel to the Golgi apparatus, which modifies, sorts, and packages them into vesicles for delivery. Meanwhile, single membrane sacs called lysosomes act as the disposal system, using digestive enzymes to break down waste and recycle damaged cell parts. Running this entire operation requires massive amounts of energy. Mitochondria break down glucose through cellular respiration to release chemical energy. In plants, specialized plastids like chloroplasts use sunlight to synthesize that food in the first place. Stripped of its surrounding tissue, a single cell is a complete automated city, with its own internal transport networks, power plants, and waste management systems. Because individual cells have physical size limits and defined lifespans, organisms rely on cell division to grow and repair tissues. Through a process called mitosis, a single cell copies its chromosomes and splits to produce two genetically identical daughter cells. Sexual reproduction requires a totally different approach. Meiosis is a two-step division that produces 4 distinct cells, known as gametes. Each contains only half the original number of chromosomes, ensuring genetic diversity in the next generation. Scientists can track this genetic inheritance directly. Indian botanist Arun Kumar Sharma developed advanced laboratory methods to study the behavior and structure of plant chromosomes, allowing researchers to accurately map how traits are passed down through these cellular divisions. Healthy cells know exactly when to stop dividing through a process called contact inhibition. They halt replication when they physically touch surrounding cells. If the DNA governing this essential control system gets damaged, the cells divide uncontrollably, leading to the formation of tumors. The continuity of life relies on the strict, highly regulated division of these microscopic units. When that regulation fails, the organism itself is compromised. We now understand these cellular mechanics so deeply that we can build them. In 2010, scientist J. Craig Venter and his team chemically synthesized an entire bacterial DNA sequence from scratch and inserted it into a host cell, successfully forcing it to grow by entirely artificial instructions. Decoding the intricate structure of the living cell gives us the ultimate power to reprogram the mechanics of life itself. To test your biological reasoning and master complex scientific concepts, head over to edunudge.in and try our 5-step nudge system today.